1. Okay, so today there's no visual. You're just going to see a picture of my screen. But right now what I'm doing is I actually have the test in front of me. And I am just going to go through the questions that I have and pick some important things from each question that I think you should focus on when you're studying for your exam. So of course this does not take the place of reviewing your PowerPoints, reading your chapters, going through all the online homework. But right now I'm just kind of narrowing some of it down for you. You still need to go over all of those materials. And I'm just picking out keywords and things you should focus on. So, you know, as you're going through this podcast, I would listen to a question or some type of association I tell you that you should know. And maybe some strategies are to pause it and then write the answer down for yourself each time I ask a new question. Other strategies may be after you've studied all the materials, maybe just listen to this podcast, pause after each question, and be able to say it, right? So when you can actually explain something to someone else or in your own words without having to refer back to something that's on paper, then you can be confident that you truly know that material and you'll be able to remember it. Also remember, I do know that this course can be very difficult for some people. You know, just biology in general is a completely different language, so there's many barriers to being successful in biology. Many of you have never even heard of these terms. This is the first time you've heard of this stuff. So do the best that you can. The reason that I include a case study in your exam grade is because that's something you can do outside of class. You can seek all of your own resources. You can ask questions about it. You can work in groups for it. And so that is the portion of the test where I can really capture what I consider to be higher level learning, where you're actually applying some of this information. And that's probably going to be the things that you remember about this class. You're not going to remember the multiple choice test. But with that said, you know, just as any general introductory class, there's a certain amount of material that has to be tested for just, you know, factual information. So, you know, if you don't score really high on the multiple choice test because you're not a great test taker, not a big deal. Don't freak out. Remember that those of you who score really well because you're great test takers or you've already taken biology or you just have a fantastic memory, then, you know, you're outside of class experience, so your extended learning, you may not have to do any of those, right? But those of you who are not great test takers, it may be one of your weaker areas, you may just have to put in a little more time than the person next to you, right? So you may need to do some extra extended learning activities, or, you know, maybe you need to study for 5 hours and the person next to you needs to study for 30 minutes. So don't worry about what other people are doing around you. You just need to really focus on what you need to do to get the grade that you want in this class. So I'm going to start off, again, just I may do some word associations or something, but as I'm looking through the test, I'm going to pick out some main points. So the first thing you need to make sure you know is we talked about macromolecules. So be able to identify all the macromolecules as well as the monomers or the individual units that make up these larger macromolecules. So for example, amino acids make up proteins and things of that nature. So be able to go through all of those monomers versus polymer association. You should also be able to list examples of each one of those macromolecules. So, you know, what are some examples of a polypeptide? Or what is an example of a lipid? Nucleotides make up what polymer? Things like that. You should also be able to list the common bonds that we talked about in class, define them, give an example. So the difference between an ionic bond versus a non-polar covalent versus a covalent bond versus a hydrogen bond. So we talked about all those bonds, be able to basically just define them and know the difference between them. We also talked about 3 domains and also in addition all the kingdoms within them. So be able to identify those domains. Where are the prokaryotic cells found? Where are the eukaryotic cells found? And examples of each one of them. Also, what is the difference between a prokaryotic and a eukaryotic cell? Back to macromolecules, be able to list when we talked about, what did we talk about? Oh, carbohydrates. What is the difference between a monosaccharide, a disaccharide, and a polysaccharide, and be able to give examples of each one of those. We looked at the periodic table quite a bit. For the atoms that we talked about in class, you should be able to identify or use the periodic table or information that I give you in the question to make predictions about the proton number, the electron number, and the neutron number, right? So the atomic mass number. You should know what the atomic mass number tells us, what the atomic number tells us, and how to calculate or how to find the subatomic particle numbers for each one of those. What is the difference between saturated and unsaturated fatty acids? There's a structural difference there, as well as a physiological difference there, how it acts in your body. When we talked about the cell structure, or eukaryotic cell structure, we listed all of these different organelles. So be able to identify the organelles and also what is the function of each organelle. When we spoke of pH within the course, you should know the difference between an acidic versus a basic pH in relationship to the hydrogen versus the hydroxide ion concentration. So which one has the most hydrogen ions? Which one would have the most hydroxide ions? And so on. What is a buffer? What is the study of biology? So how is biology defined? Again, structure and function. There is a bunch of questions about the function of your organelles within the body, or within each cell. Also, know the organelles. Remember on those PowerPoint slides, some of those organelles are found in both animal and plant cells. Some are only found in animal cells. Some are only found in plant cells. So be able to list those, the differences between those two cells. We looked at the ecosystem, how energy is incorporated into an ecosystem. Remember we talked about the sun as being the ultimate form of energy, and we talked about producers and consumers. So be able to look at that diagram and identify the parts of it. Defining, again, hydrogen bonds. What would define a hydrogen bond? You should know the difference between a dehydration reaction and a hydrolysis reaction. So how is water used in each one of those? Is it released or is it used? Is energy released or is it used? What is the role of those? Which one breaks down polymers? Which one builds polymers? We also talked about, when we were talking about, well, I'll just mention, know what Tay-Sachs disease is. Which organelle is involved? What happens? We briefly spoke about an example of an isomer. Should know the example we talked about in class. And what is the main difference between those isomers and the names of them? Only the one we talked about in class. Where are chloroplasts found? What are their roles? When we talk about proteins, we spoke about amino acids being monomers of proteins. And we also talked about the structure of those individual amino acids. So know those structures. What are the same in the amino acid structures, and then what is different? What makes each amino acid different? We also briefly talked about charges on atoms. So, and we went over a few examples. So be able to tell me, if an atom has a +1 charge, what does that mean? If it has a -2 charge, what does that mean? And just a hint is, remember, it tells us the ratio between protons and electrons. So take a minute to figure that out. Be able to identify numbers as more acidic or more basic on that pH scale that we looked at between 0 and 14. Defining isotopes, what subatomic particles are involved in isotopes. Be able to identify examples of hydrolysis and dehydration reactions. So In each one of those macromolecules, we talked about the individual units that made up those polymers, and we also talked about the fact that hydrolysis and dehydration reactions occur in all four of those macromolecules. So if I were to say, in a hydrolysis reaction, what products would be made if I were to break down a triglyceride? Or if I were to break down a polysaccharide, what would your individual components be? No, an easy way to identify a carbohydrate, there's a specific ratio that we talked about in class. So you should be able to identify that ratio and what those atoms are. What is a peptide bond? Where would I find it? What does denaturing mean in association with a protein? Which macromolecule is highly dependent on shape? We talked about different levels of organization, so be able to identify an example of each level. So remember we said there were molecules which made up cells, which made up tissues, and tissues made up tissue systems and examples. And we went over a few questions where there were some examples of each one of those levels, so review those. Again, organelle with function question here, make sure you study those. Proteins have three different necessary shapes, so the primary, the secondary, and the tertiary. And then there's the quaternary structure, which is optional. What are all of those structures? What do they look like? What do they entail? We spoke about water for quite a bit, why it was essential to life. So make sure you know those properties of water like adhesion, cohesion, polarity, solvent, and know an example of each one of those. Examples of polysaccharides. The structure of the mitochondria is unique. How is it unique, and what's the purpose of it? Reactivity of atoms. What does an atom need to be full and happy? How could you predict if an atom's going to be very reactive, or how would you know if an atom's going to be unreactive or very stable in reference to those valence electrons? The difference between a bacterial cell and an animal cell's genetic information. Where is that information found, and what the location of that information, where is it housed? What are those structures called? Now some examples of steroids. Explain the statement that DNA provides an underlying commonality between all living things. So what do we all have in common? What does a bacterial cell versus a plant cell versus an animal cell have in common? What's the difference between a positive and a negatively charged atom? What makes up fatty acids? Are they polar? Are they nonpolar? What makes up a steroid ring? What structure would identify it as a steroid? Be able to count atom numbers. So there's one or two questions where it would ask you, in this equation, so remember where are the products found and where are the reactants found, be able to identify that in an equation. And then you should also be able to count the number of atoms. So based on that big number in the front and that subscript number, you should be able to calculate individual atom numbers. So how many hydrogens are on the reactant side, or how many oxygens are on the product side? Give an example of an element where they found. Know the difference between a theory and a hypothesis, the main differences between those two. How do they differ? What is the process of hydrogenation? What does it entail? Components of a nucleotide. What components make up a nucleotide? And what monomers are they? If you put a bunch of these nucleotides together, what would you make? Again, monomer-polymer pairings, those types of questions, you should be able to pair the monomer with the polymer in the example. We went over an example of a hierarchy of organization. So review the question that we went over in class as far as putting lowest organization to the highest or vice versa. Again, basic versus acidic solutions. So in a basic versus an acidic solution, which one has a higher hydrogen ion concentration? Which one has a greater hydroxide concentration, or vice versa? Be able to predict the complementary strand of DNA. So if a DNA strand reads GGA, TTCGA, what would the complementary strand be of DNA? Characteristics of your Animalia, your Plantae, your Archaea, your Bacteria, and your Protista kingdoms. So identify, I think that this was a challenge question we went over in class, or that you had. Make sure you can identify those defining characteristics. How did you organize them? If I have two atoms and I tell you that they are both carbon, what subatomic particle would you look at to know that they are both carbon? The outermost electron shell in an atom is called a what? What type of subatomic particles are found there? And specifically, what are they called and what is their role in the atom? What is the difference between deductive and inductive reasoning? In an example, the difference between cells, tissues, organs, and organ systems. So which make up which? Which order do they go in? Why is water polar? Very important question. We looked at some really nice pictures of electron microscopes. So scanning and transition electron microscopes, just look at those examples. There's only one question about it, but make sure you just look at it and be able to tell me, like, why would I want to use a scanning electron microscope? Or if I showed you a picture, why would this be considered a transition electron microscope? If I were a scientist and I was trying to identify the genetic material in this organism as DNA or RNA, which compounds would I want to label in order to be able to see the difference between DNA and RNA? So what are some of those differences? And last, ice or water is very unique in that the solid form of ice actually floats in the liquid form. Explain to me why.